Good afternoon, everyone. My name is Yogita Warkhade, and I'm from Michigan Technological University. Today, I will discuss how microbes in the Great Lakes help degrade, break down hydrocarbon pollution. Okay, so we know that Great Lakes is one of the world's largest freshwater system, and it provides 20% of Earth's surface freshwater, and it is essential for drinking water, biodiversity, and economy. But it is threatened by hydrocarbon pollution, mostly comes from industrial discharges, oil transport, oil spill, and urban runoff affect biodiversity. This slide shows crude oil and its components. Crude oil is complex and it mostly consists of hydrocarbon. And hydrocarbon is classified as alkenes, cycloalkanes, and aromatics, and they are hard to degrade. So at the bottom, there is this biodegradability hierarchy. So alkanes are easy to degrade, but aromatic hydrocarbons and resins and asphaltenes are very difficult to degrade. So, how microorganisms play a role in this bioremediation? The microbes, they work together with different enzymatic capabilities and break down hydrocarbons. And this happens in two ways. One is sorry. So this happens in two ways. One is aerobic biodegradation, and second, anaerobic biodegradation. If so, here in I don't know. It's okay. I can see arrow. So in aerobic biodegradation, certain types of enzymes like monooxygenases and dioxygenases and start degrading hydrocarbons, and it converts to alcohols. And with the set of certain enzymes, it gets converted to fatty acids. In anaerobic biodegradation, microbes works differently. They use fumarate addition and other certain types of enzymes and produce R-succinyl CoA or R-aromatic CoA. And then these products, fatty acids, R-succinyl CoA, R-aromatic CoA, enter into common metabolic pathways like B oxidation, beta oxidation, and TCA cycle, and then the hydrocarbon gets converted into harmless products. So why should we study this in the Great Lakes? Till now, most of the studies focus on marine system, and the freshwater systems like Great Lakes are underexplored. And the Great Lakes research use control microcosm. So this study focuses mostly on natural sediment communities. So our objective was to use metagenomics to explore the diversity and functional genes of hydrocarbon degrading microbes in the Great Lakes sediments, and to investigate both aerobic and anaerobic pathways, and also to understand how microbial communities vary across location and support bioremediation potential. Now I'll talk about method section. This map shows the sample location. So MI141, straight 1, straight 3, superior 1, superior 4. These are the 5 location. The samples were collected by using this Van Veen grab tool, and after collection of the sample, this microbial samples, DNA got extracted, and DNA and the sequence, the DNA sequenced. Once it's done, we downloaded raw reads, this raw data from the Joint Genome Institute, and did further computational analysis. So we followed this pipeline. First, we checked quality of raw reads and cleaned the DNA reads by using tool FastQC and Trimomatic. Once it's done, the short sequences built into longer sequences and group them into genomes, which is called as MAGs, metagenomic assembled genome, by using tool MegaHit MetaBat2. And these MAGs, we did we did the quality checks by using tool checkm. After that, to find and label genes in the genomes, we used proca. For the taxonomic identification, we use GTDB-TK, and the functional gene identification, we used HMM model, which is can't hide, specifically for hydrocarbon degrading genes. We further did gene quantification and normalization by using HT-seq count. Now, I'll talk about result. This figure shows microbial community composition in the Great Lakes sediment, basically who lives in the Great Lakes. So from first figure, we can clearly see that proteobacteria mostly dominates in all the samples, followed by actinobacteria. And in some of the samples, there is a presence of uroviricota, which are viruses. The second figure is the genus composition, and here it clearly shows that Pseudomonas is mostly dominant in all of the samples, followed by Streptomyces. There is also presence of Burkholderia, which is also very common for, it is well known for hydrocarbon degradation. Next figure, from this figure, it clearly shows that Mi141, straight 1E, and superior 1A had highest abundance of aerobic hydrocarbon degrading genes, and those are mainly from desulfobacteriota and methylomirabiliota. In contrast, superior 4B shows abundance of anaerobic hydrocarbon genes, mostly from chloroflexota and acidobacteriota. This figure shows the distribution of aerobic hydrocarbon degrading genes and anaerobic hydrocarbon genes. And from the first figure, it clearly shows that the Mi141 had the highest aerobic hydrocarbon degrading genes from Pseudomonodata, desulfobacteriota, and chloroflexota. And in the second figure, superior 4B clearly shows that more anaerobic genes. And from both the figures, we can clearly shows that Pseudomonodata dominates in all. This is the figure is the gene counts of hydrocarbon degrading pathways. And from this figure, we can clearly see that Mi141 had the highest TPM counts, which dominates, mostly dominated by aerobic aliphatic genes. And superior 4B is that shows relatively more anaerobic genes, which indicates oxygen limited conditions. Next, we did bind and non-bind analysis. Bind is we grouped sequences into microbial genomes, and non-bind we examined all sequences. So we did this comparison, and we found out that aerobic aliphatic genes were the most abundant in both bind and non-bind data. Also, we found out that Mi141 and straight 3F had the highest total gene counts, and superior 4B had the lowest in all categories. So, our study concluded that proteobacteria, especially Pseudomonas, dominated hydrocarbon degrading communities. It also showed that Mi141 had more aerobic genes, while superior 4B showed higher anaerobic gene abundance. We also performed statistical analysis. I don't have those slides. I didn't add those slides. But we performed statistical analysis, and with that, we found out that hydrocarbon degrading gene abundance and diversity varied by location. And all of these results supported the site-specific bioremediation strategies for effective hydrocarbon pollution. I would like to thank my advisor, Dr. Stephen Tickman, and crew from NOAA who assisted with the sample collection, Nagisa Mahmoodi for sharing those DNA samples, and GLBIO 2025 for the travel fellowship. Thank you so much for your attention, and I would like to, if you have any questions.
Hi, I may have missed this, but were there differences? So in this set of HMMs that you used.
Sorry, I cannot.
Sorry, in the set of HMMs that you used.
HMM, yes.
Yeah. So what is the kind of like diversity that's represented in those protein families, and do you think that there's, you know, other protein families that are not in that set that would be, you know, kind of like that maybe distinguish these communities from other locations in the Great Lakes that might lead you to other enzymes that are important in doing this chemistry?
Yeah, so there are like other different protein families, but we only focused on hydrocarbon degrading enzymes, and we looked for those enzymes only.
Got it.
Yeah.
Okay. My question was more like, you know, are there other, you know, there are other, you know, kind of, I think the word is like desmolytic, right? Like carbon-carbon bond cleaving enzymes that may not be in your set that may be kind of like novel. Are you interested in kind of maybe looking at other uncharacterized protein families in addition to the ones that you already looked at?
No.
Okay.
No.
Okay.
Yeah.
Thanks.
We only look for, those strains. Yeah.
Thank you.
Yeah.